Once transcription is complete, the mRNA transcript can move into the translation phase. In prokaryotic cells, such as bacteria, this process is fairly straightforward. The transcript has been produced directly in the cytoplasm, so all it needs to do is find a ribosome attached to it and translation can begin. In eukaryotic cells such as our own, however, there's an extra processing step that has to happen to the transcript before it can leave the nucleus. Special molecular caps and tails are going to be added to the ends of the transcript. This is done for three reasons. First, the addition of these molecules helps protect the transcript from enzymes that might otherwise chew them up. Second, these caps and tails help the transcript leave the nucleus through one of those big complicated nuclear pores. And third, these caps and tails help the transcript attach to a ribosome so that translation can begin. Two different kinds of alterations are made. To the 5' end, a molecule known as GTP is added. GTP is a chemical cousin to ATP. It just has a guanine attached instead of an adenine. To the 3' end of the transcript, a long series of adenines will be added. This is known as a poly-A tail. And this should be poly-A tail, not ollie-A tail. The middle of the transcript is also altered through a process known as RNA splicing. In this process, sections of the transcript known as introns, which we also used to call junk DNA, are actually snipped out of the transcript. And the remaining sections, which we call exons, are going to be stitched together into a final transcript that will go on and be translated and used to build a protein. Now, why in the world would you want to chop up your transcript like that? Aren't all of the letters of the sequence important? Well, I want you to think of that transcript as being like a recipe for a set of proteins. You can actually make more than one protein from the letters used in each transcript. This would be sort of like looking at a recipe book and maybe just looking at the chapter on soups. Think about all the ingredients you can put into a generic soup. You can put chicken or beef or shrimp or fish, all kinds of vegetables, different kinds of broths, even weird stuff like beer and wine and noodles. Now, if you put all of those ingredients into one big soup, it's probably going to taste really weird because it's kind of overwhelming. So what you do instead is you look at the recipe and you decide which ingredients are going to stay in, these are the exons, and which ones you're going to leave out this time and maybe use next time. In this way, the cell can alter the transcript. It can alter the recipe a little bit to make a specific kind of protein from that recipe. And the next time it decides to use that gene, it might snip different introns out so that it can alter and make a different protein the next time. This gives the cell a lot of control, and it allows it to make multiple proteins from a single gene recipe. Here's another way to think of it. Let's say you're doing a presentation for class and you want to use in your presentation some diagrams that are in your textbook. Now, you don't want to snip diagrams directly out of your textbook, right? Or you'll mess it up. The same thing goes for DNA. You don't want to chop up the DNA or you'll cause permanent mutations. So what you do instead is you make Xerox copies of the diagrams you want from your textbook. That's like transcribing this gene. Now we can snip this transcript up however we want and we won't damage the original DNA. And so that's what the cell's going to do. Maybe this time the cell decides to keep exons 1, 2, 3, and 5, and it's going to snip out 4. Or it could snip them in a different pattern and make a different transcript. Here it's gotten rid of transcript or exon 3, and it's decided to keep exon number 4. When these two transcripts are translated, they will result in two different proteins with two different functions. So this process gives the cell a lot of control over which proteins are being made at which times for specific purposes. There are several different types of molecular machines that can complete the splicing process, including spliceosomes and some RNA pieces called ribozymes. Spliceosomes are really cool. Let's take a look at them. What the spliceosome will do is it will take that transcript and it bends it so that the intron is bent down in a loop. And then it can snip the introns from the exons at specific points, and then it bends and it puts the two together so that these molecules will join into a single transcript. Here we have the intron section that was cut out. Now that the excess introns have been snipped out and the caps have been put on the transcript, that transcript can travel out to a ribosome and begin translation. Translation includes three phases: initiation, elongation, and termination. And yes, these steps do have exactly the same names as the phases of transcription, which is convenient for memorization purposes. However, keep in mind that different stuff is going to happen during these phases, so they're not identical, they just have the same names. Let's take a look at each phase. Ribosomes are the site of translation, so translation cannot occur without a ribosome being present. Ribosomes may be present in the cytoplasm, just floating around free, or they may be found attached to the rough ER. Ribosomes are complexes of RNA and proteins kind of mashed together into this shape. Let's take a look at its structure. Ribosomes consist of two subunits, a large subunit on top and a small subunit on the bottom. The job of the small subunit is to hold a piece of mRNA in place so that the ribosome can read the mRNA. The large subunit has three sort of slots built into it that we call binding sites. These sites are known as the E site, the P site, and the A site. The E site stands for exit, so you'll want to know that term. The P site and the A site also stand for things, but you don't have to worry about their names. Now, what are those slots for? Well, these are sections where tRNAs are going to fit into the ribosome, and they're going to be carrying the amino acid ingredients that we need to make the protein. tRNAs, or transfer RNAs, consist of a long strip of RNA that's been folded up on itself. And there are certain sections that are complementary, so it can hydrogen bond with itself to form this kind of T shape. There are two other regions of the tRNA that are very important: the anticodon on the bottom and an attachment site for an amino acid on the top. Now remember that tRNAs are like the assistant to the chef. They are responsible for bringing specific ingredients over to the ribosome in the right order and at the right time, and they put those specific ingredients in the right place to make the protein that's trying to be made. Now, since we're making protein, the ingredients we're talking about are amino acids. There are many different tRNA molecules, and each one has a specific amino acid attached to its top. So it's responsible for transporting that amino acid and putting those amino acids in the right spot in that protein. Regions of each tRNA, known as an anticodon, help that molecule figure out where it needs to drop off its specific amino acid. Remember that the tRNA is just made of a strip of nucleotides that's been folded into a shape. And it's been folded such that these three little nucleotides on the bottom stick out. That's known as an anticodon. What that anticodon is going to do is it's going to match with a specific codon in the mRNA transcript. So here we have CUC. That would match with the sequence GAG, or GAG. That's kind of fun. So if that mRNA sequence contains the sequence GAG, this tRNA can bind with it, anticodon to codon, and that tells it that it needs to drop off its amino acid in that specific spot. Here we can see another view of this process. This is the mRNA transcript, and you'll notice that the transcript can be broken down into groups of 3 nucleotides that we call codons. Now we're already kind of in the middle of translation in this diagram. So the ribosome has already added a couple of amino acids to the beginning of this protein. And it added those specific amino acids based on these specific codons that were in the sequence. Now this next little tRNA comes along. It's carrying an amino acid on its top, and it happens to have this anticodon CUC. It just happens to find its matching codon in the sequence, GAG, GAG. And so what that tells the tRNA is that it needs to drop off its amino acid and attach it to this growing chain. And so that tells it to leave that amino acid in that specific position within this growing protein. Let's put all these great molecular machines together so we can see translation in action. The first phase of translation is initiation, and as in initiation of transcription, what we're trying to do in this phase is assemble the machinery. The machine we're trying to assemble is called the translation initiation complex, and it's going to involve the ribosome, the mRNA, and the tRNA all fitting together in a certain pattern. First, we have the small subunit of the ribosome, and what it's going to do is latch on to the mRNA transcript. It's going to position that mRNA transcript so that the start codon is directly in the middle. It's going to be in the P site when the large subunit of the ribosome joins up with all of this. Now notice what's attached to that start codon. On top of the start codon, we have the first tRNA. The start codon always has the sequence AUG, so the first tRNA that's going to hook up with it will always have the anticodon UAC. And if you remember your translation chart, AUG always codes for the amino acid MET, or methionine. So this particular tRNA is carrying methionine on top. MET is always the first amino acid that's added to a protein chain. It's not always necessary for the protein's structure and function, so sometimes it's removed later, but we always start building a protein with MET. Once those pieces of machinery have assembled, the large subunit of the ribosome is going to come in and it's going to slide into place over the rest of this machinery. It'll position itself so that the first tRNA is in the P site and so that the first amino acid on top is kind of sticking out of this slot on the top. At this point, the translation initiation complex is complete and we're ready to go into phase 2. The second phase of translation is known as elongation. What's being elongated in this phase is the amino acid chain. So we're building the actual protein at this point. Now this diagram kind of starts in the middle of elongation, not at the beginning, but we can walk through it. We're going to start with the translation initiation complex being all constructed. There's a tRNA in the P site with an amino acid attached to the top. The next thing that's going to happen is that another tRNA, whichever one matches up with the next codon, is going to come along and it's going to fit into the A site. So now we have a tRNA in the P site and the A site, and they both have their specific amino acids sitting on top. Now we have to do a little shuffling around. The tRNA that's been sitting in the P site was hanging on to the amino acid chain until now. But notice what it's done. It's passed that amino acid chain on to the next tRNA that's sitting in the A site by attaching its amino acid chain to the amino acid on top of the tRNA. Now that it no longer has an amino acid on top, that tRNA can leave. So what will happen is that all of these components will scoot over by one codon, by one series of 3 nucleotides in the mRNA. That's going to put the original tRNA in the E site. And remember, E stands for exit. So this little guy is going to leave. He's going to go find another amino acid and attach it to the top so it can do the job again. The tRNA that is now in the P site has the amino acid chain attached to its top. This cycle will now repeat over and over and over again to continue growing that amino acid chain. It'll start with a tRNA in the P site, another one will join it and bring its little amino acid with it. The amino acids will transfer from the first tRNA over to the second tRNA. Here it's attached to the second tRNA. That allows everything to scoot over by one codon so that that original tRNA can leave, leaving the amino acid sequence attached to the tRNA that's now in the P site, and it starts over again. This will continue until the stop codon is reached. Once that stop codon slides into the ribosome, the third phase, known as termination, can begin. Now, the stop codon doesn't actually code for an amino acid, so it won't match up with a tRNA carrying an amino acid. Instead, it will combine with this molecule known as a release factor. Release factors actually carry a little molecule of water on top. So what that will do is attach a little molecule of water to the amino acid chain, and that causes all of the machinery to fall apart. So here the machines have fallen completely apart. The ribosome can go off and grab another mRNA to translate. The tRNAs can go get refilled with amino acids and do their jobs. Sometimes this mRNA is reused and retranslated to make more of the same protein, or sometimes it gets broken down and recycled, depending on the needs of the cell. The end of the termination phase marks the end of translation and the end of the entire gene expression process. At this point, several different things can happen to this little amino acid chain that's been created. Sometimes it can fold up into its final shape and go off and just do its job straight away. Other times, that amino acid chain represents a small polypeptide, and that will need to be added together with other polypeptide units to build a bigger protein. Sometimes these amino acid sections also need to go to the Golgi apparatus, and as they travel through the Golgi apparatus, they finish folding, and they have other things added to them, such as carbohydrates, that are necessary for doing their jobs. At any rate, that protein has been created, so it can go off and do what it needs to do. All right, you guys, you made it. You made it all the way through the process of gene expression, from DNA to finished protein. Make sure you fill out your notes thoroughly and bring them to class. Jot down any questions you have, and we'll work with these molecules in class together.